we have the insulin signaling pathway, and Jacob is part of our group, but he's quarantined, so he could not be here for our presentation, but he helped with the PowerPoint. And then Perry, Isaac, John Murray. Yep.
We also helped.
The origin of the insulin. Insulin is secreted from the cells in your pancreas, which is right here on this diagram, and the pancreas detects increased blood glucose levels in the duodenum, which is the first part of your small intestine right here, and it will begin secreting the insulin to regulate the blood glucose levels. Inside the pancreas, which is the picture here, there are groups of cells called the islet of Langerhans, which are mostly made up of the alpha and the beta cells, which we can see here. And when blood glucose levels increase, the beta cells will detect it and begin releasing insulin into the bloodstream. At the beginning of reception, insulin will act as the ligand and bind to the alpha subunits of the insulin receptor, which is the RTK. Once this happens, the beta subunits of the insulin receptor will become activated, forming a dimer, which creates a conformational change in shape, and the use of ATP to phosphorylate its tyrosine residues. After phosphorylation, the pathway will move into the transduction phase, which starts with the phosphorylation of IRS1 and 2.
Okay, for transduction part 1, the first step is over here, where the beta subunits of the RTK will phosphorylate the insulin receptor substrate, otherwise known as IRS, right here. So there's multiple types of IRS, but typically we'll be dealing with just IRS1 or IRS2. After IRS1 is phosphorylated, the enzyme PI3K will bond with IRS, as seen here. This bonding is done through PI3K's P85 subgroup. Once this bond is formed, PI3K will phosphorylate PIP2 to PIP3, as seen there. Now you'll also see that there's a P10 there. That P10 is used for regulating the pathway, which we'll get to later. The PIP3 will increase in concentration, attracting two proteins, PDK1 and AKT, towards the plasma membrane. The PIP3s kind of recruit those proteins. So once the PDK1 moves towards the plasma membrane, it will become activated, and upon activation, it will phosphorylate AKT. Once AKT is activated, it will phosphorylate AS160. Now, AS160 typically is not phosphorylated. When it's not phosphorylated, its job is to prevent the translocation of a GLUT4 vesicle into the plasma membrane. So the GLUT4 vesicle wants to go into the plasma membrane, but AS160 typically just stops that. Once it's phosphorylated though, AS160 allows the GLUT4 vesicle to translocate into the plasma membrane, becoming a protein embedded in the plasma membrane. So now that the GLUT4 protein is embedded in the plasma membrane through the translocation, it will allow for glucose to pass in and out of the cell. Typically glucose will come into the cell to start glycolysis or power other cellular functions, and glucose will leave the cell through the same protein if the blood sugar content level of the person, or I guess animal, has a really low blood sugar content level, which is typically why people get insulin shots for diabetics, because their blood sugar content level is low.
Okay, so in this part we have transduction part 2, and so AKT is going to do more than phosphorylate AS160, so it will also phosphorylate GSK3, and this is important because GSK3 will normally inhibit GS through phosphorylation, and GS is just glycogen synthase. And so this is important because when AKT inhibits GSK3, GS will stop being inhibited. So you're basically inhibiting an inhibitor, which means the inhibition will stop. So as previously stated, GS is just short for glycogen synthase, and glycogen synthase is an important enzyme in glycogen synthesis, which is a process that converts unused glucose to glycogen. So it basically is going to turn the glucose into, it's going to store it for later energy use. So in summary, in this pathway, the cellular response is not only the moving of glucose and starting up glycolysis through the GLUT4s, but it will also allow glycogen synthesis to happen. So this is where that P10 that John Murray talked about is going to come into play. So this is the insulin pathway regulation. So one way in which the cell regulates the insulin pathway is through the protein P10, and this protein will actually dephosphorylate IRS, keep the pathway from continuing. So it's used to regulate the concentration of activated IRS. And another way the cell regulates the insulin pathway is also through P10. And this protein will also dephosphorylate PIP3, and this results in the recruitment of PTK1 and AKT stopping, so you won't get any so the AKT will not be phosphorylated on PTK1, which will ultimately halt the translocation of GLUT4 to the plasma membrane. And a third way in which the cell will regulate the insulin pathway is through the production of P70S6K, which is right there. And so besides phosphorylating GSK3 and AS160, AKT will also indirectly, so it doesn't directly pass a phosphate group to this next thing, but it does it through multiple steps, and it will phosphorylate mTORC1. And once mTORC1 is phosphorylated, it will then phosphorylate its downstream target, P70S6K. And this is important because these P70S6Ks will then go back to the beginning of the pathway and phosphorylate IRS through negative feedback inhibition and stop it from continuing the pathway. And this is used to help regulate the insulin pathway. And that is it.
Any questions? Nope.
No questions? Okay, we did that
Thanks.
perfectly.
Thanks, Ms. Young.